Hi, this is Mrs. Taylor doing a short video on the differences between homologous chromosomes and sister chromatids and tetrads and all these wonderful vocab terms that get very confusing. So to start with, DNA is found in the nucleus, normally in a loose format called chromatin. But when the cell is going to divide, whether it's mitosis or meiosis, the chromatin condenses into chromosomes. And that's what we see when we're looking at slides of cells in various stages of cell division. In this model, the chromosomes are being represented by the different colored pipe cleaners shown here. So on each individual chromosome, there's going to be sets of instructions, and those instructions are called genes. And the genes are then used by ribosomes to make various proteins to control all the different functions within the cells. So the genes are very important with it. Those genes are found on these different chromosomes. In a human, we would have 23 pairs of these chromosomes. I have 3 pairs shown here in this example. So we have a long pair, a medium-sized pair, and a short pair. With these chromosomes, we'll look at our medium-sized pair here. Each of these chromosomes has the exact same set of genes on them. So here we have the same genes. So that's basically the same set of instructions. If it helps you, you could think of these instructions as like recipes in a cookbook. And so each of these chromosomes is like a cookbook, and it has a whole bunch of recipes on it. These 2 chromosomes are the exact same size and the same shape. They would have centromeres found at the same location. They're the same chromosome. They're not identical to each other, but they are the same chromosome, the same sets of information. We call these homologous chromosomes. So back to the cookbook analogy, these would have the same recipes. They would have recipes for chocolate chip cookies and for peanut butter cookies and oatmeal raisin cookies and brownies and cake. So it would have all the same recipes, but the ingredients in those recipes might be a little bit different. So for example, this recipe up here might be for chocolate chip cookies, but this recipe over here is also for cookies, but they're oatmeal raisin instead. And so it's the same recipe, but the ingredients are slightly different. The end result is going to be different. So your homologous chromosomes have the same set of instructions, just slightly different versions of those instructions. And that makes them homologous. So my 2 short ones here are homologous to each other. They have the same instructions, slightly different versions. And my 2 long ones, also same thing. And it's represented by the color of these beads. So the pink stars are representing that gene, that information. And because they're both pink, it's showing that it's the same instructions, the same recipe. In actuality, this might be eye color, blue eyes, brown eyes, or hair color, blonde hair, black hair. Okay, so that's what we would be looking at in a human with it. So we have these homologous chromosomes. Before the cell can divide, all of the DNA has to be copied. So it goes through the synthesis phase, and it has to be copied exactly. So now we have all of this DNA copied exactly on these 2 different chromosomes here. So the two pink chromosomes are exactly the same. They are completely identical to each other. Every single nucleotide is the same. Same thing here for our teal ones. Everything on it, every single adenine, thymine, cytosine, guanine is exactly the same order. We call these sister chromatids. So sister chromatids are identical. If you want, you could think of them as identical twin sisters. Everything about them is exactly the same. So these two are sister chromatids. These two are sister chromatids. But our homologous chromosomes, I don't have room to write it down here. So these two are our sister chromatids. These two are homologous chromosomes. So the teal and the pink are homologous chromosomes. Same information, slightly different version. Oatmeal raisin, chocolate chip. But our two sister chromatids are identical to each other. Chocolate chip, chocolate chip. Everything about them is the same. So you have to distinguish between sister chromatids and homologous chromosomes. The pairs are homologous chromosomes. When we have all four of them together, so during prophase 1 of meiosis, you're going to see the sister chromatids and the homologous chromosomes all together. This structure of all four of them together is called a tetrad. So here's our long chromosomes as well. Okay, so you have your identical twin sisters and their homologous chromosomes. All four chromosomes together form a tetrad. So your tetrad are the two sister chromatids and plus your two homologous pairs or chromosomes. So you have 4 total chromosomes. That's where you get tetrad from. So in meiosis, we talk about tetrads, especially in meiosis 1. Mitosis, you do not see the tetrads at all. They never form, so you don't have to worry about them in mitosis. That is the distinction between sister chromatids and tetrads and homologous chromosomes. One last thing that tends to confuse people is in terms of terminology. All of these are considered chromosomes. Okay, so this one by itself is a chromosome. This one by itself is a chromosome. So each one is a chromosome. Each chromosome has a centromere on it. When the two identical chromosomes are together, they're still chromosomes, but to help distinguish that they are identical to each other, the term chromatid is used. So you have your two sister chromatids, which are made up of 2 chromosomes. After they have separated and they're in different cells, We call them chromosomes again. So it's just a little terminology that confuses people. Sister chromatids, or just chromatids, really are chromosomes as well. Hopefully this short video helps.